outdoor series of histology made easy with diagrams. I have prepared for you a diagram, a drawing that will probably be helpful in putting together things that were not connected, which helps a lot to have clarity on the subject. Here we have depicted a simplified version of the central nervous system histology, the peripheral nervous system, and also the effector cells. We'll start with the central nervous system. And just to begin with, I'll remind you that the central nervous system is covered by a layer of bone in the head and in the spine. And in this diagram, it is stained orange, just for simplicity. We'll go back to the underlying layers. Maybe I should just mention here that the yellow band corresponds to the dura matter. We'll go a little bit, expand our knowledge on the meninges later. So just get to talk about the brain itself. You know that when you cut a brain, you have an area that are gray matter. That corresponds to the neurons. That's where the neuron bodies are located. And the white matter it could be at the periphery, it could be not at the periphery. It varies depending. This is not an anatomical section, which has fibers that are myelinated, as are myelinated those that are out, some of them that are outside the CNS. When you start putting your eyes into the main cell, which is a neuron, I have only painted here one single neuron. We have probably about 100 billion of them, perhaps less, but it's about around that number. Anyway, you can see, take a look, an idea, you can see that the cytoplasm of the cell is basophilic. It's blue in most cells. And obviously the nucleus is going to be also basophilic, and the chromatin is more or less somewhat dispersed. The nucleolus is large, and just to begin with, we have to understand that the largest cells in general of the CNS are the neurons, and the other smaller cells that are more numerous are the glial cells. Okay? So if you start looking at the neuron, you see that there are two types of projections, cytoplasmic projections. The dendrites, which are sensitive, that receive messages, receive input, and the axon, which may stay within the CNS or may go out of the CNS to significant lengths. Now, when you start looking at the neuron itself, you kind of start a little bit disappointed that we probably thought, I thought at the beginning that the neurons will have something really much different inside the cytoplasm. No, sorry. We have a rough endoplasmic reticulum. We have a Golgi apparatus, which usually may extend up to the dendrites to a certain extent, but do not extend to the axon. You have mitochondria, however, that are all over the place. Mitochondria are in the perikaryon or cytoplasm around the nucleus, in the dendrites, in the axons, and even in the synapses near the muscle. So it is mitochondria are everywhere. Except you have to remember that the mitochondria that are in the axon are just filamentous, very elongated, a little bit different, but still are mitochondria. Now the language of the CNS is a chemical language, and the contacts of one axon to another neuron is on the synapses, is via the synaptic or terminal button or in the synapses. In other words, the axon dilates. It contains secretory vesicles containing different substances such as acetylcholine, norepinephrine, glutamate, dopamine, and others, which will be received by, in other words, have contact with receptors in the postsynaptic membrane and have inhibitory or excitatory effects depending on the situation. So, for example, here we have an axodendritic synapse, an axosomatic synapse, and an axoaxonic synapse. So, now in general terms, we can say that the neurons are not by far the most numerous cells, as I said before. The glial cells are. The glial cells are supporting functionally and mechanical cells that have those functions, and that appear all over the place in the white and in the gray matter, and are of 4 types, if you have to remember: the astrocytes, the oligodendrocytes, okay, the microglia, and the ependymal cells. I'll try to be, I'll give you a few concepts. You probably already know about them. The astrocytes are maybe of 2 types, the protoplasmic astrophytes or the fibrous astrocytes, depending if those that are in the white matter tend to be more slender, more rachitic, should we say, and we call the fibrous astrocytes, while the other ones are called the protoplasmic astrocytes. The astrocytes are all over the place. Their nuclei are small, smaller than that of the neurons, and they actually have so many projections towards, have synapses with the neuron. It has food processes, which are extensions around the basement membrane of the vessels that penetrate into the brain from the area of the subarachnoid space. So we're dealing with a situation in which the vessel is totally covered by the food process of the astrocytes, and we have the blood-brain barrier. In other words, certain molecules cannot penetrate the brain in view of the fact of this barrier. In addition to that, the astrocytes extend upwards, or laterally, to be in touch and to cover, if you want, the lower aspect of the basement membrane of the pia mater, which is here. So in other words, the lowest, the thinnest and most lowermost meninges is totally covered by the processes from the astrocytes. Now, when you look at the brain, it's kind of disappointing because you can quite easily recognize the neurons because of the fact that I saw they're basophilic, big nucleus, etc. But when you look at the glial cells, you see a meshwork of protoplasmic extensions that cover most of the brain, if you want. They seem to, and then you cannot really quite figure out where the astrocytes, for example, are present, unless you use special stains, like Ramon y Cajal did in the early 1900s, or we can use immunoperoxidase for GFAP, glial fibrillary acidic protein, that depict precisely the location of every astrocyte. Now, astrocytes, as well as the other glial cells, are exceedingly numerous, probably 10 times as numerous as the neurons, perhaps in the number of close to 1 trillion. But they are in different locations. I'll say a few things about the microglia. The microglia is not easy to detect. We have to do some special stains like immunoperoxidase for CD68, because these cells belong to the monocyte macrocytic system. In other words, they don't belong to the brain until the last stages of pregnancy, in which they enter the CNS, and then they go around like police trying to find areas of infection or inflammation, and then they transform into really more phagocytic cells, which pretty much are the functions of a leukocyte inside the CNS. Now, before I go into the oligodendrocytes, I'll say something about the ependymal cells. You'll have to remember when you did some neuroanatomy that the brain has cavities, like the lateral ventricles, which are quite big, the fourth ventricle, the third and fourth ventricle, and the ependymal canal in the spinal cord. Those cavities are not denuded. They are covered by an epithelium. And that epithelium is about cuboidal or low columnar with cell junctions, but does not have basement membrane. So it's kind of a special kind of epithelium without basement membrane, which reminds me the other epithelium, which is not really an epithelium, is the synovium. The synovial surfaces of the body do not have basement membrane and do not have a true epithelium too, just to remind you. Now, within those cavities, you see that there are vascular proliferations lined by epithelium, which are the choroid plexuses. The choroid plexuses are different locations within those cavities, and they are the site of formation of the CSF. The CSF, cerebrospinal fluid, will penetrate into the subarachnoid space and surround the entire, and flow around the brain and inside, around the spinal cord and inside the spinal canal. And finally, they are going to be enter the Pacinian or arachnoid granulations, and the CSF is going to go into the large veins that are in the dura. That's pretty much the way the CSF flows, and it is an important thing to remember. Now we're getting close to the area of the axons in the white matter, and you can see that the axons are not denuded. They are. That's why the white substance is white. It is covered by, we call myelin, and myelin is nothing else but multiple layers of membrane that wraps around a segment of the axon, and within or outside the CNS. Inside the CNS, we have one of the groups of types of cells called the oligodendrocytes. The oligos are misnomer. The name implies that those cells have very little in the way of cellular projections, and that's not true. They have up to 50 projections, and they are able to surround different axons and different levels. For example, each of the projections acts as a spatula. Like I would say here, this would be an axon, and the membrane of the oligodendrocyte wraps around the membrane around the axon, and the axon is totally protected by this myelin layer. As you know, the membranes are composed mostly of lipids, so this is there's a high content of lipids in the white matter. Now, I think we can go to the peripheral nervous system, and you'll see that there are types of cells that, there are different types of fibers. Some of them are larger, more important. Some are probably less important or smaller in size. But all of them are protected by one cell that is not part of the CNS. Its origins are from the neural crest. And that is the Schwann cell. I just wrote it down for you, the Schwann cell. The Schwann cell wraps around the fibers, protecting them like gold, if you want, because they are so important. One after the other, one after the other. Of course, there are two ways of wrapping around or protecting a fiber or an axon. One would be forming multiple layers of membrane, like if you put your hands inside the Schwann cell and start wrapping around, you have multiple layers of membrane. exactly or in a way similar than what happens with the oligodendrocytes. Now there's between Schwann cell and another one, we have the nodes of Ranvier. That is the name it has. I wrote it down for you guys. The nodes of Ranvier. And also, in your books, you'll find the funny name of the incisures of Schmidt-Lantermann, which actually are oblique areas of the myelin sheath in which cytoplasm is preserved, and it is believed that those areas are helpful for the metabolism and the function of the myelin sheath. Now, this arrangement is also quite important from the functional point of view because the impulse jumps from node to node. That way the impulse is extremely fast in those nerves in which, for example, voluntary nerves in which that speed is needed. Smaller axons are also protected by the same cell, the Schwann cell, and the axons are embedded partially or totally in the cytoplasm of the Schwann cell, but there is no myelin, there is no membrane around them. That is something to remember. I'll just remember something I'll go back, but before I forget, you have heard that in the brain there are these brain tumors, and I'll just try to tell you that since the most important, the most abundant of the tumors comes from the glial cells, and mostly the tumors from the CNS are the astrocytomas. Unluckily and sadly, the brain produces tumors, and those are, whether they are malignant or benign, sometimes is not quite important because those tumors can extend and we cannot know exactly how to excise them because we don't have the precise borders. Now, regarding to the Schwann cells, we do have also tumors like schwannomas, or also neurofibromas that are composed totally or partially of Schwann cells. That's something I just wanted to tell you. Now, before we go to the nerves, let's see, the axons usually, when it comes to the effector organs, when it comes to the skeletal muscles, the axons branch in different branches and form motor end plates in different skeletal fibers or muscle fibers, cells. And that's important because one neuron, we could say, is going to control a certain number up to hundreds of muscle fibers, or only a few depending on the importance of that muscle. For example, the eyes, the external muscles of the eyes maybe have only a few branches because those are very, very precise, and not, for example, those muscles of the leg may not be as precise. So, and over here, what we have here is almost a synapsis, but in each of the branches of the axon is covered by a extension of the Schwann cell until the end. And the neurotransmitter that is secreted here is acetylcholine, which will produce a liberation of calcium, an impulse that will go into a liberation of calcium, and all the sequence that you have learned when you studied skeletal muscle contraction with troponin, tropomyosin, etc., etc. So this is the contraction of the muscle. Now, before going into the autonomic nervous system, well, let's say something about the nerves. When you see a section that is cut longitudinally or in an oblique fashion, it is quite characteristic that the fevered fibers are going in an ondulant, in a very, very wavy fashion, and interspersed are a few nuclei corresponding mostly to the cells of Schwann, to the Schwann cells. Also, some fibroblasts may be present. When you cut a nerve in a transverse fashion, you see that the axons are a little bit not easy to see because the myelin is mostly fat, and it's difficult to quite clearly see the entire sequence of a fiber. But you can see that we have connective tissue around or forming fascicles or within around the nerves themselves. So now what is the reason for this arrangement? You probably already know. I don't have to tell you. When you have to play basketball, if you didn't have this arrangement, you probably would have a lesion of your nerve or one of the brachial plexus nerves. So this is a protection mechanism for stretching that is quite useful. Regarding the autonomic nervous system, there is a difference between the sympathetic and the parasympathetic systems. I'll just tell you right away. The sympathetic has a series of ganglia that are paraspinal, and you can see them in your anatomic diagrams very clearly. There is a synapse there, and then the postsynaptic fiber close to the viscera, you see the heart, the bowel, or the uterus, whatever. So, however, the parasympathetic does not have the ganglia that at this level, but the ganglia are very close or within the viscera, for example, in the case of the bowel, or even adjacent, very close to the heart. So this is something to remember. Finally, before I forget, I will mention something about the meninges. I told you that the yellow corresponds here to the dura mater. In the head, around the brain, the dura mater is tightly attached to the bone, so that in an autopsy you will probably have difficulty removing the dura mater from the bone. However, I'm just reminding you that in the spinal canal, the dura mater is separated from the spinal bones, from the vertebral bones. And there is an epidural space full of veins that is used for anesthesia or different situations. So there's a different situation between of the dura mater between the skull and the spine. That is important. Now, below the, I'm putting here a little bit that below the dura you have a membrane that's fibrous, that is thinner than the dura, yes, but thicker than the pia, which is very thin. The arachnoid has projections, fibrous projections, to the pia mater and forms that way a space, a subarachnoid space, which is filled with cerebrospinal fluid and also contains vessels that surround the brain or surround the spinal cord, which are going to penetrate and are going to be partially covered or totally covered by the extensions from the astrocytes. But anyway, this is the way the arrangement. The pia mater is lined by cells that are mesenchymal type cells that are so flat that almost look like fibroblasts. They do have a basement membrane and do have the projections from the pia mater from the astrocytes to the pia mater. I think with this, I'll have to just probably point out here that from the subarachnoid space, you have these projections into the dura, which are the arachnoid granulations, which are going to be the site of absorption, if you want, of the CSF into the veins that are located in the dura mater. I hope this graph diagram is useful to you, and I see you next time. I'm glad to see you. Bye-bye.